Hello, everyone. My name is Gray Powell, and today I will be discussing how we can uncover the mechanisms of neuropsychiatric diseases using zebrafish. Zebrafish are great candidates for research for neuropsychiatric diseases as they share 70% of their genome with humans. They're also sustainable, cost-effective, easily maintained, and they develop rapidly. Previous genome-wide association studies have associated a specific gene, the ZNF804A gene, and its enhancer with two neuropsychiatric diseases, schizophrenia and bipolar disorder. ZNF804A functions in the brain in neurodevelopment, in DNA transcription. It regulates the gene expression in the brain, and most importantly, it's responsible for functional connectivity, or generally how the brain is interconnected and is able to transport messages. The ZNF804A gene and its enhancer concentrate in the prefrontal cortex, the striatum, and the cerebellum, all of which are essential neurostructures. How the ZNF804A enhancer works is that the enhancer region lives upstream of the downstream ZNF804A gene on the DNA strand. So as the RNA polymerase is moving across the enhancer region, creating mRNA, the enhancer is telling the RNA polymerase to make more of the target gene or the downstream gene. Previous GWAS have specifically associated a deletion mutation at the SNP or single nucleotide polymorphism RS1344706. This deletion mutation has caused a frame shift in the enhancer region DNA, which will therefore affect the production of downstream ZNF804A gene. While GWAS have implicated this mutation with causing the development of bipolar disorder and schizophrenia, scientists still do not fully understand the genetic and biological mechanisms of this mutation. I wanted to understand how this mutation worked in the development of schizophrenia and bipolar disorder in the brain. So first, I had to generate a line of mutant zebrafish. To do this, I used CRISPR-Cas9 gene editing technologies to insert this mutation into the fish's DNA. Then I allowed for the fish to grow for 6 days post-fertilization, allowing some of those essential neurostructures to develop. And then I isolated the brain tissue. From the brain tissue, I extracted RNA, synthesized cDNA, and used this cDNA in RT-qPCR, which would allow me to quantify the expression of the ZNF804A gene relative to a specific housekeeping gene in the brain. The results from my study will allow us to better understand how enhancers interact in the brain, in the body, and in genetics. It will also allow us to develop a more comprehensive drug therapy for these two diseases and develop better genetic screening, as well as further develop psychotherapies so that we can truly comprehensively treat and help those suffering from schizophrenia and bipolar disorder.